Hello, everyone. Thank you for being here. My name is Logan Thornton, and I'm joined with,
Natalie Getzi.
and we're students in Dr. Peg Riley's lab. Today, we're going to be talking about the battle against bacterial speck and how we can protect our tomatoes. What will this presentation cover? We'll begin with a discussion about what bacterial speck of tomatoes is. We'll talk about the issues associated with antibiotic treatments, also the importance of friendly bacteria in plant growth. Then we'll introduce an alternative to the current bacterial speck treatments, specifically what we did and what we found, and we'll end with our conclusion and future studies.
So, what is bacterial speck? It's a disease which impacts tomatoes, and it's caused by the gram-negative bacterium Pseudomonas syringae PV tomato. This is one of 60 pathovirus, or pathogenic variants, known to infect plants. The bacteria attacks the tomato through its seeds, or through openings in the stem, root, or fruit part of the plant. It leaves black speck-like dots all over the fruit, as well as yellow halos on its leaves. P. syringae thrives in moist environments, as well as warm temperatures, but it can also survive during the winter on plant debris, weeds, and seeds. Its spread is impacted by overhead watering, rain, and sprinkler systems. Additionally, cross-contamination can impact spread by handling infected plants and then healthy plants with hands or gardening tools. So how does this disease of tomato impact the economy? In 2017, the tomato market value is determined to be $1.67 billion, which is not surprising considering its culinary and nutritional value. Tomatoes and tomato products contain folate, vitamin C, and potassium, making them a great part of the human diet, as well as the second most common food behind potatoes on the market. P. syringae stunts tomato growth, and it prevents the fruit from reaching maturity, which reduces yield. Once a fruit is infected, it cannot be saved. Thus, this leads to a 75% crop reduction and millions of dollars wasted. Some current treatments for P. syringae PV tomato include streptomycin and oxytetracycline, both of which have rising rates of antibiotic resistance among bacteria and have been used since 1958 and 1972, respectively. Copper sulfate is an additional treatment which has also recorded resistance. Scientific studies have found that current spray treatments against bacterial speck of tomato are inefficient. What are some off-side effects of antibiotics? Antibiotics used on plants and animals are washed into waterways, where bacteria thrive and can transfer resistance through plasmids. As mentioned previously, aquatic environments allow for bacteria such as P. syringae to reproduce and spread resistance. Antibiotics are broad-spectrum killers, meaning that they kill both bad and good bacteria. This has severe consequences because they can harm plant growth promoting bacteria, which are considered good bacteria. What are these PGPRs? Rhizobacteria, which are species that live in the soil and surround the plant roots and participate in mutualistic relationships with the plant. This means that they both benefit each other. The rhizobacteria can actually help reduce the level of disease. Some ways in which they can do this is through pathogen exclusion, or outcompeting for resources, nutrient acquisition, toxic metal uptake, nitrogen fixation, hormone production, and toxin production. By producing a killing agent without harming beneficial microbes, collateral damage can be minimized to ensure plant growth, increase crop production yield, and ultimately produce healthy tomatoes to meet the global market demands.
Now that we've heard some ways that these rhizobacteria are important, let's look specifically at the tomato rhizosphere. Here are two figures from Dong et al.'s 2019 paper. Each figure is broken down into the rhizosphere, the microorganisms in the soil near the roots, the phyllosphere, the bacteria on the plant above the soil, and the endosphere, which is the microbiota within the plant itself. In figure A, the y-axis shows the Chow-1 index, which is a measure of bacterial diversity, or how many species are present. The x-axis shows multiple sampling locations within each part of the tomato microbiome. You can see a much higher level of microbial diversity in the rhizosphere compared to the other locations. This diversity is vital to proper plant health. We even see differences in microbial diversity within the human microbiome and on different parts of our own body. And if you're wondering, one of the sites on the human body with the highest diversity is in our gut. Figure B shows the relative abundance of several genera of bacteria and other microorganisms. These red arrows point to genera of bacteria that were used in our study, which we will discuss shortly. You can see that the rhizosphere is not dominated by one particular genus, like over here, rather several genera are abundant. So how do we protect these rhizobacteria, or these PGPR? Well, we're interested in bacteriocins. Bacteriocin is a very broad category that encompasses diverse and highly specific protein antimicrobial defense weapons. These weapons usually are targeted to closely related species. They have unique and varied mechanisms of action. For example, nisin, which is our model bacteriocin in the lab, is organic, non-toxic, and actually likely in the yogurt you ate this morning. Nisin kills bacteria by binding to the lipid 2 membrane within the gram-positive peptidoglycan cell wall. This inhibits peptidoglycan biosynthesis, but nisin can also insert several copies of itself into the membrane and form a pore, which effectively kills the cell. While there is the possibility of resistance, the threat is much lower because loss of these conserved binding receptors, such as lipid 2, incurs a loss of fitness to the cell. So hopefully you can start to see how bacteriocins are of high interest for use against plant pathogens in agriculture, because they essentially are targeted missiles to kill pathogens and preserve the beneficial PGPR. And this was exactly the goal of our study, to screen for bacteria that produce bacteriocins against Pseudomonas syringae PV tomato. How did we do it? Well, we took advantage of the fact that we can induce bacteriocin production through competition for limited resources, such as when they're grown together on a nutrient agar plate. In this figure on the left, you can see that we have a nutrient-rich agar plate with a lawn of bacteria spread over it. 2 different isolates were spotted directly on top of the lawn. Isolate 1 produced a zone of inhibition and effectively inhibited the growth of the lawned isolate, while isolate 2 did not produce a zone of inhibition. This was repeated for 217 bacterial isolates against themselves and each combination. Isolates ranged across 19 genera and 44 species. 52 isolates were of agricultural plant pathogens, while 74 isolates were classified as plant growth promoting rhizobacteria. Within this all-by-all assay, we specifically looked at the interactions of these 217 isolates against 12 lawned isolates of Pseudomonas syringae. With an overarching goal of finding an alternative treatment for tomato bacterial speck, we were interested in finding the most effective producer of inhibition against the most P. syringae lawns. In this figure, you're seeing the percent of the total P. syringae lawns inhibited by the best producing isolate within each species that was found to produce. Numbers in parentheses represent the total number of isolates tested within the species. You can see 2 producers that showed high percentage of inhibition. One is Enterobacter aerogenes, and the other is an isolate of alkalogens. When we look at these 2 producers specifically on the plate tested in triplicate, we can see that these isolates are producing an effective and highly consistent zone of inhibition. Now, let's compare the number of pathogenic lawns able to be killed to the percentage of PGPR isolates inhibited. Enterobacter aerogenes inhibited 75% of the P. syringae lawns and 21% of the PGPR. Alkalogens, however, inhibited 67% of the P. syringae lawns and only 9.4% of the PGPR isolates. So what does this mean? We know that there is an inhibitory compound that is produced consistently through cell contact, likely due to competition for resources, which activates bacterial defense mechanisms. Both Enterobacter and alkalogens are classified as PGPR. We know that one way that PGPR promote growth is through antimicrobial production. Perhaps we can find the secrets of fighting plant diseases through studying the symbiotic interactions within the rhizosphere. This figure was created with data obtained from our assay, as well as from relevant literature. It contrasts the P. syringae killing to PGPR killing by the standard treatment and our proposed treatment. Something that wasn't mentioned previously is that there has been success in combining several treatments, such as copper and streptomycin, together, and because they often kill more pathogens together than either can alone, they are said to show synergy. There have also been documented reports of bacteriocin synergy when different peptides are introduced into a single treatment. If we combine the 2 best producers we found, we can raise the total number of P. syringae isolates inhibited from 75% to 93%, because each producer inhibited slightly different strain. This producer cocktail would only raise the total PGPR inhibited to 25%. You can see how this is much lower than the reports of copper and streptomycin on PGPR, which also show much lower efficacy against P. syringae due to tolerance and resistance gene transmission. In conclusion, 2 producers of inhibition against P. syringae PV tomato were identified at the isolate level out of 217 potential candidates. They showed effective inhibition of the P. syringae and low levels of PGPR inhibition. These results suggest a treatment for bacterial speck that may not only kill the pathogenic bacteria, but preserve the plant growth promoting bacteria, which may even increase tomato crop yield. Some limitations of the study are that we tested a limited number of species, several of which included only a single isolate. Also, true drug discovery often requires screening of thousands of potential candidates to find a suitable producer. Future studies will focus on the identification of the inhibitory compound. This will be done through protein purification, sequencing the genome of the bacterial producers to search for the inhibitory gene. Once we know what is actually doing the killing, then we can move on to testing on the tomato plant. Here are our references. Thank you and take care.